All right, welcome back to the deep dive. Today we are shrinking right
Oh,
down
down.
to the most fundamental building blocks of life, the cell.
Mm.
Think of this as your express ticket to really understanding what makes up every living thing around you, from bacteria to whales.
And hopefully, yeah, helping you ace that science exam, too.
Exactly. We've been looking closely at a whole stack of GCSE science revision guides.
We have.
Yeah.
Pulling out the key stuff you really need to get your head around cell structure function.
And what I find fascinating, how these tiny units get specialized for amazing jobs.
Yeah, the adaptations are incredible.
Our mission here is to get super clear on the essential parts of different cells, what they do, and how their structure fits their function perfectly. We want you feeling really confident about this.
Definitely. Because when you look closely, I mean, with microscopes, you find this astonishing complexity. It's all
Yes.
there, hidden away.
Tiny wonders. Let's unpack them.
Let's do it.
Okay, so the guides are really clear. All living things, made of cells, but
But they're not all the same. Not by a long shot.
there's this massive dividing line, isn't there, separating life into two main camps.
Yep. Eukaryotic and prokaryotic. That's your starting point.
Eukaryotic and prokaryotic, okay.
Yeah.
What's the core difference?
It really boils down to internal organization, how things are arranged inside.
Right.
Eukaryotic cells, think of them as having compartments, little rooms inside. They have a proper nucleus.
Control center.
Exactly. Housing the DNA. And they have other structures, organelles, wrapped in their own membranes.
So membrane-bound organelles, like little walled-off sections for different jobs.
Prokaryotic cells, on the other hand, much simpler, much more basic layout.
Oh, nucleus.
No nucleus at all. Their DNA, their genetic material, it's just floating, loose in the cytoplasm.
Oh, okay.
And they don't have those membrane-bound organelles we just mentioned. No mitochondria, no chloroplasts, none of that complex internal structure.
So who's in which group? Examples.
Easy ones to remember. Animal cells, plant cells, they're your classic eukaryotes. Bacteria, they're the main example of prokaryotes. Single-celled, much simpler.
Got it. Animal and plant eukaryotic. Bacteria prokaryotic. Now, seeing these things, you need help. Microscopes come up a lot.
Oh, yeah. Essential.
We've got light microscopes and electron microscopes mentioned. What can you see with each? What's the difference?
Good question. So light microscopes, they use light, obviously, pass it through the specimen.
Okay.
They're great for seeing the bigger picture, whole cells like animal or plant cells. You can usually see the nucleus, maybe some larger structures like mitochondria if you stain them right.
But there's a limit.
There is. It's about resolution, not just magnification, how much detail you can distinguish. Light waves themselves are relatively long, so you can't resolve things that are really, really tiny and close together.
Like trying to see tiny details with blurry vision.
Kinda, yeah. You hit a wall with light. Electron microscopes, though, use beams of electrons.
Electrons, okay.
And electrons have much, much shorter wavelengths. That's the key.
Shorter wavelengths. Better resolution.
Massively better resolution. So you see the ultrafine structure, the details inside mitochondria or chloroplasts. You can see really tiny things like ribosomes.
Ah, the protein factories.
Exactly.
Yeah.
And you can see the detailed structure of those much smaller prokaryotic cells, the bacteria. You need that electron power for the fine details.
So light scope for the overall layout, electron scope for the intricate machinery inside. Makes sense.
Yeah. It's how we really learned what's going on in there.
Yeah.
So let's dive into those eukaryotes first, starting with, say, an animal cell. Pretty familiar.
Okay, animal cells. The guides list the main parts. First up, cytoplasm, called jelly-like.
Yeah, it's basically the filling, mostly water,
Yeah.
but it's packed with dissolved stuff, nutrients, salt, enzymes, and crucially, loads of chemical reactions happen right there.
It's not just padding, it's where the action is.
A lot of it, yeah.
Yeah.
A busy workshop. Then the nucleus.
The control room we mentioned.
Yep, contains the DNA, the genetic blueprint. It directs everything the cell does by controlling protein production. Got its own membrane, too, in eukaryotes.
Right. Boss and blueprint. Next, the outside boundary. The cell membrane. Described as selectively permeable.
That's a key phrase for your exams. Selectively permeable means it chooses what gets in and out.
Like bouncer at a club.
Huh. Sort of, yeah. A very specific bouncer. Controls nutrients coming in, waste going out, holds the cell together, and maintains the internal environment.
Okay. And mitochondria. The powerhouses.
That nickname really sticks, and for good reason. They contain enzymes for aerobic respiration.
Aerobic, meaning using oxygen.
Yes, it's the main way cells release energy efficiently from glucose, using oxygen. That energy powers everything the cell does.
So if a cell does a lot of work, like a muscle cell,
It'll be crammed full of mitochondria, absolutely packed, More mitochondria, more energy.
Makes sense. And finally, for the basic animal cell kit, ribosomes.
Tiny little things.
Yeah.
Really tiny. You need that electron microscope to see them clearly.
But important.
Vital. They are the sites of protein synthesis. They build the proteins, following instructions from the DNA in the nucleus. Proteins do almost everything, build structures, act as enzymes.
So the workers that build everything based on the nucleus's plans?
Pretty much. So recap animal cell basics: cytoplasm, nucleus, cell membrane, mitochondria, ribosomes.
Got it. Now, plant cells, also eukaryotic, so they share that basic setup.
They do. They have all those parts. Cytoplasm, nucleus, cell membrane, mitochondria, ribosomes, same basic functions.
But plants are different from animals, obviously, so they must have extra bits.
They definitely do. Some really important additions that are crucial for plant life.
Okay, what are the key plant extras?
Number one, cell wall. This is outside the cell membrane.
Outside, okay.
Yeah. It provides structural support, protection, rigidity. In plants, it's made mainly of a strong carbohydrate called cellulose.
Cellulose, that's what makes plants sturdy, like wood.
Essentially, yes. Gives them shape and stops them bursting if they take in too much water. It's like an external skeleton. Quick note, though, bacteria also have cell walls, but they're made of different stuff, not cellulose.
Good distinction. Okay, cell wall. What else?
Chloroplasts. These are amazing. They're green because they contain chlorophyll.
The pigment for photosynthesis?
Exactly. Chlorophyll traps the sunlight energy. Chloroplasts also have the enzymes needed to use that energy, plus water and CO2, to make glucose, the plant's food.
So the plant's personal food factories, powered by sunlight.
Couldn't have put it better. Essential for life on Earth, really.
And the last big difference the guides mention.
The permanent vacuole. Plant cells usually have one really large central vacuole.
A big storage sac.
Kind of. It's a membrane-bound space filled with cell sap water, sugars, salts, waste products. Its main job is maintaining turgor pressure.
Turgor pressure.
Yeah, it fills with water, pushes outwards against the cell wall. This makes the cell firm, turgid. It helps support the plant, keep leaves, and stems from wilting.
Ah, so that's why plants droop when they don't have enough water. The vacuole shrinks.
Precisely. Animal cells might have small temporary vacuoles sometimes, but nothing like the big permanent one in plants.
Okay, so let's summarize eukaryotes. Animal cells, cytoplasm, nucleus, cell membrane, mitochondria, ribosomes, plant cells, all of those. PLUS, a cellulose cell wall, chloroplasts, and a large permanent vacuole.
Perfect. That covers the eukaryotic side.
Now let's jump across that divide. Prokaryotes, focusing on bacteria.
Right. Single-celled, prokaryotic, key feature, no nucleus.
So their DNA just floating around.
Pretty much. Their main genetic material isn't wrapped in a nucleus. It's typically a single, large loop of circular DNA, just there in the cytoplasm.
Called the chromosomal DNA?
Correct. But there's often more.
The guides mention smaller circles of DNA, too.
Yes. Plasmid DNA.
Yeah.
Little extra rings of DNA also in the cytoplasm.
And these are interesting.
Very. Plasmids often carry extra genes, like genes for antibiotic resistance. And crucially, bacteria can copy these plasmids and pass them to other bacteria.
Wow, even different types of bacteria.
Sometimes, yeah. A major way they share genetic information and adapt really quickly. Explains a lot about how antibiotic resistance spreads.
That's huge. Okay, so free-floating DNA, the main loop, and these shareable plasmids. What else makes up a bacterium?
Well, they still need the basics. They have cytoplasm, like eukaryotes. They have a cell membrane controlling what gets in and out.
Right.
And they have ribosomes to make proteins. They're simpler, but they still need to build things.
Okay. Cytoplasm, membrane, ribosomes, free DNA. Any other unique bacterial bits?
Many have flagella. Singular is flagellum.
Like little tails.
Exactly. Whip-like structures, sometimes one, sometimes many. They rotate or move back and forth to propel the bacterium, let them swim around.
Briny propellers.
Basically, yeah. And like plants, they have a cell wall outside the membrane for protection and shape.
But not cellulose.
Definitely not cellulose.
Right.
Made of different materials like peptidoglycan in many bacteria. Same function, structure, and protection, but different chemistry.
Okay, so bacterial checklist. Cytoplasm, cell membrane, ribosomes, chromosomal DNA, loose loop, plasmid DNA, small loose circles, cell wall, not cellulose, and often flagella for movement.
That covers the key parts. The guides often have useful comparison tables, which are great for revision.
Yeah, let's quickly hit those main comparison points between prokaryotes, bacteria, and eukaryotes, animals, plants. First up, size.
Big difference. Prokaryotes are tiny, typically 0.2 to 2.0 micrometers. Eukaryotes are much bigger, 5 to 100 micrometers usually.
So potentially 100 times bigger. Wow.
Yeah, it's a major scale difference. Next, genetic material.
The core difference again.
Yeah, eukaryotes. DNA in a nucleus, linear chromosomes. Prokaryotes. DNA free in the cytoplasm, main circular chromosome plus plasmids.
No nucleus prokaryote. Nucleus eukaryote, easy rule. What about organelles?
Eukaryotes have all those membrane-bound ones, mitochondria, chloroplasts in plants, vacuoles, others. Prokaryotes, none of those.
None at all.
No membrane-bound one. They only have ribosomes, which aren't membrane-bound. Both types have ribosomes.
Interesting. So bacteria manage energy without mitochondria.
They do. They use different pathways, often involving structures in their cell membrane, simpler systems. And finally, cell division, how they make more of themselves.
Different processes.
Very different. Eukaryotes use complex processes like mitosis to carefully copy and divide their chromosomes. Prokaryotes, being simpler, just replicate their DNA loop and split in two. It's called binary fission.
Binary fission. Simpler, faster reproduction.
Generally, yes.
That comparison really helps nail the differences. Okay, moving on. One of the coolest ideas is specialization.
Absolute critical for multicellular life like us. Cells don't just stay generic, they differentiate, they become specialized.
Meaning
Meaning they develop specific features, shapes, structures, internal bits that make them really, really good at one particular job. Structure is adapted for function.
form follows function at the cellular level. The guides give great animal cell examples. Let's look at the sperm cell. Its job, fertilize the egg.
Right. So how's it built for speed and delivery? It has a long tail, or flagellum, packed with protein fibers that contract. That makes it swim.
Swimming needs energy.
Loads of it. So the middle section, right behind the head, is crammed with mitochondria. Power packs right next to the engine.
Clever design. And the head itself.
The very tip has a structure called the acrosome. It's like a cap full of digestive enzymes
Enzymes to do what?
to break down the egg's outer layers. When the sperm reaches the egg, it releases these enzymes to drill through, allowing the sperm nucleus to enter.
A biological drill bit. And inside that head.
The haploid nucleus, haploid meaning it has half the normal number of chromosomes, ready to combine with the egg's half. And yes, sperm are tiny, produced in millions.
Contrast that with the egg cell or ovum. A much bigger role initially.
Huge role. Its job is to be fertilized, yes, but then to provide everything needed for the first few divisions of the embryo.
So it's packed with supplies.
Exactly. Its cytoplasm is rich in nutrients, yolk, or other stored food molecules. That provides the energy and building blocks for the early embryo. It's also much larger than a sperm, one of the biggest cells in the body. Fewer are made.
And it also has a haploid nucleus waiting for the sperm's half.
Correct. And its cell membrane does something amazing after fertilization.
Oh, yeah.
As soon as one sperm gets in, the egg cell membrane changes chemically, instantly becoming impenetrable to any other sperm.
A defense shield goes up. Why?
To prevent polyspermy being fertilized by more than one sperm, that usually leads to an incorrect number of chromosomes and the embryo wouldn't survive. So first sperm wins, then the door slams shut.
Incredible mechanism. Okay, one more example. Ciliated epithelial cells. Found where?
Lining surfaces like your airways, your trachea, and bronchi.
And their special feature is in the name, cilia.
Yep. Tiny hair-like projections sticking out from the cell surface. Lots of them.
And they move.
They do. They beat back and forth in a coordinated rhythm, like tiny oars creating a wave.
What does that wave do?
In your airways, it sweeps mucus upwards. Mucus traps dust, bacteria, other debris you breathe in. The cilia constantly move that mucus up towards your throat so you can swallow it or cough it out. Keeps your lungs clean.
So the cells themselves stay put, but their cilia do the sweeping.
Exactly. The cells are anchored, forming a lining, and the cilia provide the movement. A fantastic example of structure, perfectly suited to function clearing your airways.
It's amazing how these tiny structural adaptations allow for such vital functions.
It really is the essence of multicellular complexity. Cells specialize, taking on different forms to do different jobs efficiently, all working together.
So, recapping our deep dive, we started with the big split prokaryotes, like bacteria versus eukaryotes, like animals and plants.
We looked at the key parts: nucleus, mitochondria, ribosomes, cell walls, chloroplasts, vacuoles, and what they do.
Then we saw how eukaryotes in particular take these basic parts and modify them for specific role specialization, like sperm cells built for speed.
Egg cells packed with resources.
And ciliated cells acting like tiny brooms.
Understanding that link between a cell's structure and its specific function is so important, especially for your exams.
Absolutely. Hopefully walking through it like this helps you visualize those parts. Maybe sketching them out, like the guides suggest, or using those keywords, nucleus, control, mitochondria, energy, ribosomes, proteins.
Whatever helps make it stick. Comparing animal and plant cells side by side is useful too, seeing what's the same, what's different, and why.
Definitely. Okay, a final thought to leave you with. We've talked about cells specialized for movement, reproduction, cleaning. Think about other jobs in your body.
Like sending nerve signals, or contracting muscles, or absorbing food in your gut.
What structural adaptations might those cells have? If you looked at a nerve cell or a muscle cell under a microscope, what features would scream, I'm built for this specific job?
How does their structure tell the story of their function?
Something to ponder. The amazing specialization that starts with these fundamental building blocks. Thanks for joining us on this deep dive into the cell.
Until next time.